This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode number 223, recorded on March 8th, 2013. Hi, everybody. I'm Vincent Racaniello, and you are listening to TWiV, the podcast all about viruses. Joining me today from Western Massachusetts, Alan Dove.
Good to be here.
I bet you'd rather be in Grand Bahamas today.
Yes, I would much rather be in Freeport today.
Did you get a lot of snow?
A surprising amount. They were calling for the forecast was all over the place, and we ended up getting 6, maybe 8 inches.
Oh, you have to shovel it out?
I was able to use the snowblower on it this time. It was not so deep that it was beyond the capacity.
Excellent. So you had your workout today then?
Yes. But it's now it's slushing. So it's a little rain and melting and big mess.
Yeah, we just had a few inches here. No big deal. It was supposed to be a lot more, I think. And now it's all gray and 1 degrees Celsius.
Yeah.
So we still have a winter weather advisory. I don't know what that's all about. Snow will continue into this afternoon or early evening. Okay, that's fine. Also joining us today from Ann Arbor, Michigan, Kathy Spindler.
Hi, Vincent. Hi, Alan.
Hey, Kathy.
You get snow out there?
No, and it's 40 degrees and sunny, so the remaining stuff is melting.
Nice.
Cool.
Yeah.
Spring is here, huh?
Yeah.
So when is spring, and is it this month or next? Next
March
month?
20th, I think. Yeah.
It's in March, huh? We're almost there.
Yeah.
Yes, go ahead.
I was just going to say, I was noticing that it's 4 degrees Celsius. So now we know that when we talk about, the 4 degree room or the 4 degrees, it's 40 Fahrenheit.
Yes.
Oh.
Huh. It's not very cold, is it? I guess.
No.
Yeah, well, it's a few degrees above freezing, right?
Yeah.
That's good enough. Ours is always broken, so it doesn't matter. Right? So, Kathy, today is an interesting day?
Yes, it's International Women's Day. And we have a number of international women in our department on the faculty. And one of them is very gung-ho about having us celebrate every year. So she sent out a memo to the whole department describing what the day is about. And both last year and this year, she arranged a dinner for the women to get together. So, yeah. It started in the early 1900s, and it may have a little bit of, at the start, may have had a little bit of socialism overtones, and it's always been much bigger in countries all around the world than in the United States. Mm-hmm. But evidently in 1977, it was formalized by United Nations. So it's March 8th every year.
So in theory, every nation should be celebrating it, right?
Right. But it's like Mother's Day or whatever. It doesn't hurt to have a special day as well.
Right. Well, actually, I would say in theory we wouldn't need this sort of thing, but we do.
Yeah. So is that true? If all things were equal, we wouldn't need it?
Yeah. I mean, you wouldn't have to have special distinctions, I am in favor these days of celebrating the work of women, but if we were all equal, we would say, oh, well, why make a point of it?
I don't know. Maybe not.
But I think in the current world, this absolutely makes sense. Sure. This is an excellent idea.
Mm-hmm.
And I think your pick has something to do with that, as we will see later.
And that was kind of coincidental.
Oh, yeah?
But it all came together, yes.
Maybe and by the way, I had bookmarked that as a potential pick, too. So you...
But maybe that's...
You took my pick.
Maybe nature timed it for today, right?
Yeah, maybe.
Who knows?
Maybe.
All right, we have some FU, quite a bit.
That's follow-up for those who have just joined the show.
First one's from James Chen, the principal author, the principal investigator on those 2 science papers we did last week. I guess Rich wrote him because he writes, "Hi, Rich. Thank you so much for featuring our work on your podcast. It was a great honor. It is really gratifying for my postdocs and students to hear their work analyzed and praised by the experts. Quick answers to some of your questions. Praise will get you nowhere." We are not the experts, but thank you. 1, C-gas can be activated by single-stranded DNA, albeit more weakly than double-stranded DNA. It's not activated by RNA. All right, we had asked that question during the show. 2, there is some C-gas in the nucleus, but the majority is in the cytoplasm, where it detects viral DNA, possibly on their passage through the cytoplasm or release from the phagosome. 3, in our vaccinia virus experiments, we used heat-inactivated virus, which can be taken up by cells and deliver DNA, but probably lost the ability to make new viral proteins that might antagonize the DNA sensing pathway. 4, malaria most likely engage this pathway. I'll just add that for the purification of C-gas, we used more than 2,015 centimeter plates of L929 cells, performed mass spec of over 100 bands by my student Xiaji Wu, which generated over 100 gigabytes of data that were analyzed by label-free quantification to identify a set of proteins that co-purify with the activity throughout the purification steps. We had to do all of these because of the very low abundance of the protein. We estimated that we needed to purify the protein from the crude extracts by about 1 million-fold to achieve apparent homogeneity, which is nearly impossible without an antibody. HeLa and 293 cells do not have this cytosolic DNA sensing pathway, i.e. C-gas and or sting are not expressed in these cells. We think this may be one of the reasons these cells are so popular for plasmid transformation. I'm altering his statement.
It's not transfection.
Depends if you're putting a virus in. Wow, 2,015 centimeter plates.
Ouch.
Yeah.
It must have taken them days and days and days to grow those up, right?
Yeah.
They had to do it in batches. And those are expensive. I mean, we don't use them anymore. They're just too much.
Well, the plates, the media,
I mean, like, the thing is...
I would say, you know, there's also the cost of the time to do the experiment, but it's a student, so...
Yeah.
The thing is, how would you know? I mean, what did they do, 100 and said there's not enough, or they just decide go right for 2,000? Or maybe they did some small batches and it didn't work. Anyway.
Well, they probably did several batches, you know, working out the different columns, and they did the next prep with the columns in the different order, and
maybe in the end it all took 2,000.
Right.
Yeah, it seems like the kind of thing you'd work up to.
It's like Albert Sabin said, he used 7,000 chimps and 10,000 monkeys to make polio vaccine, but not all at once.
Right.
Wow.
That's more than a barrel.
Oh, sure is. And when we have him on, we can use that as the title.
Oh, who's Sabin? Yes.
Yeah. I want to see if you were listening.
Yeah, that'd be nice.
Alan, can you take the next FU?
Sure. Yes, I can. Robin writes, I think we've heard from Robin before, writes about open access in an anarchy. The solution to open access is the nonviolence of anarchy, laying down the gun of coercive violence of enforcement by the state and all petty oligarchs and gangs. Sans state, there would be no copyright, no corporate charters, and therefore no corporations. Interactions would be horizontal, voluntary, non-coercive instead of vertical and coercive. An interaction cannot be deemed voluntary when it's at the gunpoint of the state, no matter how heavily cloaked. In answer to the standard response, who would build the roads, it would be the same people who do so now, but the money to pay them would not be confiscated at the state's gunpoint. And he adds a phytometazoan would be a combination of a metazoan and a metaphyton. And the nearest link I could think of was corals.
Huh. Did we talk about phytometazoans?
I don't recall.
I guess.
Dixon was trying to think of a word for the plant equivalent of a metazoan, the word would be.
Yeah, I would just add that sans state, you would solve the problem of open access because there wouldn't be any funding for basic research.
Yeah.
So there'd just be no science to publish.
I have a naive question. Does anarchy not have violence?
There's this, well, people who advocate anarchy are usually advocating a utopian version of it where everybody just gets along and cooperates and has these nice interactions. You know, it's like any utopian worldview, like pure communism or libertarianism or what have you, where somehow, you know, people are honorable and don't try to cheat the system.
It's like an academic department, in other words.
Yeah. Of course, it doesn't work because people suck, but...
Okay. Kathy, can you take anonymous?
Sure. Dear Twivers, In response to TWiV 222, which contained a large discussion about how to deal with scientific corruption, I would like to voice my own humble opinion. It is certainly true that those who discuss clear problems with the scientific process can unintentionally alert the public and give them ammunition to hinder the scientific process. Also, those naysayers, negative Nellies, also have a knack of bruising the egos of scientists who need that ego to keep them motivated in the face of the technical and intellectual challenges that face them from the time they wake up to the time their head hits the pillow. Anonymous doesn't mention the financial challenges, the grant writing challenges. Oh well. But we should certainly not dismiss those problems as not real or wacko just because we cannot think of a solution. Indeed, a huge problem in science is people not revealing their own negative findings. How many scientists must hopefully press on, repeating experiments and trying to prove hypotheses that have already been disproven in the hands of multiple people? How often do we not provide advice to people when they present their research just because we fear that we will either bruise their ego or tip our own hand and be scooped in the process? We must ultimately ask ourselves the question, are we here to build our own careers, or are we here to advance our understanding of disease and the world around us? Signed, Anonymous.
Good last sentence there.
Yeah.
I would say that we're here to advance our understanding of disease, right?
Mm-hmm.
I would say I would agree, of course, but I would add those aren't mutually exclusive.
Well, you have to build a career, right? Otherwise, you can't do anything.
Yeah, ideally, advancing our understanding of disease will build your career.
Okay.
And why do you want to build a career, Alan?
Well, to be able to pay the mortgage.
And also so that you can advance understanding of disease and propagate that knowledge, that understanding.
Do something useful for humanity.
Well, yeah, I mean, you build a career so that you can have a lab and get grants and publish. If you don't have a career, you can't do that.
Right.
The mortgage you don't need. You could live in a cave.
That's true.
Or you could rent an apartment.
Right.
Which many people here in New York City do. Next one is from Ed, who writes, Dear Vincent, Alan, Rich, Alan, Kathy, and Dick. And Alan apparently was duplicated.
Yes, I'm here twice.
No error correction there. At the beginning of TWiV 222, you mentioned the 60th anniversary of the announcement of the structure of DNA. At Cold Spring Harbor Lab, a meeting was organized to celebrate the event with a plethora of esteemed seminar speakers, including several Nobel laureates. Though the meeting concluded on Sunday, James Watson, Sidney Brenner, Matthew Meselson, and Wally Gilbert were invited to speak on Saturday evening. They recounted the heady days of the discovery, events that led up to it and followed. It was punctuated with some irreverent humor, too. But something that Matt Meselson said struck a chord with me, though he said it slightly tongue-in-cheek. He said that someone should really get this four, and by extension more people of their era, together to talk for a week and recount the way things were done, how, why, and the life of scientists back then. It reminded me of all the times you talk about getting people on before they die and their oral history is lost. Please instigate a regular feature on TWiV, TWIM, and TWIP alongside Virology 101 in which you talk to someone with such an oral history. I think it's vital. It need not be for a regularly scheduled TWI either. It could be a special episode, as I'm sure they'd be very popular. Second, with regards to the DNA gassy sensor papers. That's a good one. Gassy DNA. A real tour de force, as you said. I do share some concerns regarding how exactly some DNA viruses are sensed in the cytoplasm, but I would also contend there is still much to be understood about how gas and other purported DNA sensors interact or place separate parts in this process. The proteins DAI, RNA Pol3, AIM2, IFI16, and DDX41 have also been implicated as DNA sensors. How do their mechanism of sensing differ from each other? Do they act in concert or in different subcellular locations or compartments, induce different signaling outcomes, recognize different DNA or DNA protein complexes as substrate? If each were a sensor for one pathogen versus another, that might give a clue, but this information seems lacking compared to the vigor it was pursued with regards to sensors that recognize foreign viral RNA. So here's a pick of the week here. Maybe should we save it for later?
Sure.
Yeah.
I'll remember a group of picks about
the
60th anniversary. Yeah.
I'll copy it and put it down there. And meanwhile, Alan, can you take the next one?
Yeah. So a friend of mine on Twitter who posts as Dr. Ricky, DRRICKY on Twitter, is a TWiV fan, and he posted a few responses after the last show. He said, so it's all these are in short tweets, but I'll try and read them as if they were a letter. Listening to TWiV, yes, there's evidence of retroviruses in protozoa. The problem stems from the incorrect use of the term. Protozoa is a morphological classification, not evolutionary. At least 5 lineages are lumped in there. At least in the amoebozoa, a sister clade to animals and fungi, yes, the genomes are littered with retroviral copies. Mind you, I don't think a retrovirus has been observed in action among amoebozoa, but the tracks are there. But this is also the lineage where mimivirus was discovered.
So this needs to be reclassified by evolutionary criteria then. Is that the idea?
Yeah, I think it probably has been, but we keep using the term protozoa.
I see.
But yeah, apparently the amoebae have retroviral copies, and you had some questions about these that I don't know the answers to.
I'm wondering if they are real. So the hallmark of a retrovirus is, LTRs flanking the insertion and duplication of host sequence. I just wonder if that's been found as well, or if these just retroposons that have been popping around, by virtue of having reverse transcriptase in the genome.
Right.
But if Ricky is listening, maybe he will.
He is probably listening. Yeah.
So he's a bioinformaticist and a dicty researcher.
Yes.
Dictyostelium.
So he knows a thing or two about amoebae and sequences.
So dicty is an amoeba, right?
Yep.
And according to the current classification, it's a protozoa. Or is it amoebozoa?
I guess it's amoebazoa. Actually,
that's what he calls it. But he says it's lumped in with the protozoa.
Right.
I wondered if this was all one tweet. It couldn't have been, right?
No, it was five or 6 different tweets that split this into. So, Ricky, if you want to drop a line through the TWiV comment form, if you want to elaborate on this a bit, or you could send me to 20 or 30 follow-up tweets.
See, if we were live streaming the audio, we could say, "Hey, people in the chat room, what do you think?"
That's right.
Someday.
Yep.
Kathy, you have a follow-up.
Well, just after we watched the Condit Dam video, there was a little bit of discussion about what happened afterward. And somehow on that same page, I found a link to what happened at least 6 weeks after the explosion. And they showed that the fish were doing okay. And so far, they had the response that they wanted. So if we want to, put that in the show notes or whatever.
Sure. The next one is from Jonas, dear fellow virologist. To be fair to my European colleagues, I just wanted to give a short notice that you missed one name for IPS1, Visa Mavs, in the last episode of TWiV. Malin et al. Published the same protein in Nature in the fall of 2005, same time period as the other articles, but called it Cardiff because it is a card-containing adapter protein that recruits I-kappa kappa kinases. Cardiff. A couple of years ago at a conference, one of the authors of the Cardiff paper mentioned in a presentation that all the authors from the 4 different papers had agreed on using the name MAVS. So that's what I have done since. It at least seemed like a reliable source since he was not on the MAVS paper. Thanks for an easy way to broaden my knowledge about other viruses than the specialized field I am doing my research on. Jonas is over in Sweden. I don't know. I don't think they've settled on MAVS because I see other names being used. But thanks. I did forget Cardiff, yeah. Alan, take Ibrahim's, please.
Ibrahim writes, Dear family TWiVidy. I like that.
That's very nice.
I had a question about scientific writing, and I tried to solve it in TWiVo, and I wanted to thank you that the answer was helpful and to the point. So I would like to take a chance to encourage enthusiast virologists to try the in TWiVo system if they have any inquiries or virus-related problems. I would certainly also refer microbiologists and parasitologists to check out the in TWIMO and in TWIPO systems, too. A follow-up to TWiV 222. Last time when Dr. Racaniello asked why would the DNA sensor be in the cytoplasm instead of the nucleus, he said he was told it was because the system is leaky and some viral DNA might reach the cytoplasm during infection, and this is when the viruses are being sensed. He also said he just simply doesn't buy it. I also don't. It seemed to me that it's just pure speculation, not based on any evidence to think so. I mean, it seems that there are no experiments done to quantify viral DNA leakiness to the cytoplasm during the course of an infection, and whether this is a phenomenon that would push a cell to develop viral DNA sensors in the cytoplasm to fight infection. But anyway, knowing that people speculate without evidence, I want to take my turn. I think that maybe this sensor is not in the cytoplasm specifically for viral DNA. Maybe it's just for any DNA, and that the presence of DNA in the cytoplasm is completely prohibited. Therefore, the sensor is just there, and viral DNA is being treated as any other criminal DNA being in the wrong place.
That's a good idea.
Yeah.
Yeah.
But I still think there's something else for viruses that are in the nucleus.
Yeah, my problem with the leakiness hypothesis is it just isn't weird enough.
I think it's not elegant.
I mean, there's the elegance issue, but biology just, especially with an antiviral defense, it's usually, it doesn't seem like it's left up to chance quite that much.
Yeah.
There's probably more there.
Yeah.
Well, to me, it seems like it's a pretty elaborate system for something, if you want to hypothesize that it only happens when the viruses leak their DNA out, or in the case of adenoviruses, they've lost some of their capsid proteins and they're on the way to the nucleus, but they're still associated with some of their capsid proteins and some other proteins around the DNA. So it just seems too sophisticated to just be taking care of a leaky situation.
Yeah, that's exactly my problem.
I think maybe it would also just, it strikes me that it would be way too easy for a virus to stop that.
Yeah. The rest is not related. It's not an FU, so we'll do it later. How's that?
Okay.
Sounds good.
FU's only at the beginning. It's just an
Get the FU out of the way. Yes.
Follow-ups.
Follow-ups. That's what the FU is.
No, we would never curse on this show. We are a family show.
That's right.
All right, we have 3 papers today. And the first one is from Kathy, from a student in one of her classes, right?
Yeah, so we started a thing. We are talking about viruses in the news for the first 5 minutes of every class. And for almost the whole first half of the semester, the other professor and I have had to generate the viruses in the news topics. But recently, the students have started sending us things, so this was exciting.
This is a paper in Science Express, rather short, although lots of supplementary data. The title is Mechanism-Based Covalent Neuraminidase Inhibitors with Broad-Spectrum Influenza Antiviral Activity. And this is trying to improve on the neuraminidase inhibitors. So to review, as everyone may or may not know, there are 2 FDA-approved neuraminidase inhibitors, Relenza and Tamiflu. They work by inhibiting this glycoprotein on the virus surface that's the neuraminidase. It happens to be an enzyme that cleaves sialic acid from glycoproteins. And in fact, Ibrahim's question relates to this, so that we'll get to that later. It's just coincidental. Now, I want to point out something here. The authors say, so the neuraminidase is thought to function when the virus is being released from cells. It allows the virus to get away from cells because as the viruses are budding off, they're attaching to sialic acid receptors on the surface, sialic acid being the receptor for the hemagglutinin. So the neuraminidase cleaves it off and allows it to be released. So the authors explain this, and they say, on the basis of the notion that NA inhibitors should reduce viral spread, and I just want to point out that this is not a notion, but is in fact based on experimental results from none other than my former PhD advisor, Peter Palese, in a paper which he published in 1974, the year I was born. No, just I want to see if anyone's listening. It's in Virology, which I have a preprint of. You can't get it on PubMed. Maybe you could go to the Virology website and get it. It's called Characterization of Temperature-Sensitive Influenza Virus Mutants Defective in Neuraminidase. I remember this paper because when I was a first-year student, all the first-year students spent a week in every PI's lab for the first, I don't know how many weeks. And we'd go over papers. And this is the one he had me read and recount. It's basically, they had a TS mutant where the neuraminidase is defective at 39 degrees. And under those conditions, the viruses, you don't get viruses in the cell supernate. And when they did electron micrographs of the infected cells at the high temperature, all the virions were stuck to the surface. beautiful, a very simple paper, and it set the stage for an inhibitor, right?
Right.
So it's not a notion. And you chemists got to read the literature.
While we're quibbling over terminology,
It's not quibble. It's not terminology. It's important background. Sorry.
I did have a little objection to the title, actually. I think all drugs are mechanism-based.
Yeah. Well, they're designing these based on the mechanism.
Yes,
you know.
what they mean is that it's what I guess used to be called rationally designed drugs.
Yeah. That's in fact how the neuraminidase inhibitors were designed.
Yeah, every drug has a mechanism.
They had the crystal structure of sialic acid bound to the neuraminidase protein, and then they designed inhibitors that would mimic the way they sat in there. So the Tamiflu and Relenza, they are mimics of sialic acid. They sit in the sialic acid binding site, and they inhibit the enzyme. But these are, of course, you get resistance to these, and so there are difficulties. It would be nice to have something you didn't get resistance to and which inhibited all sorts of strains. So what they start is by investigating how the enzyme actually works, and they produce a derivative of sialic acid which has fluorines attached to it, and this essentially forms a covalent bond with the neuraminidase protein, and so this provides evidence, which they can detect by radioactive labeling, and this provides evidence that the neuraminidase, when it reacts with sialic acid, goes through a covalent intermediate. So you form sialic acid, neuraminidase covalent bond, and then it's broken later on to release it.
Right.
So then they start to say, okay, this is cool. Maybe we can use this idea to make an inhibitor that binds covalently to the enzyme, and then that would be hard to mutate because whatever it's binding to, and they have evidence that it's the tyrosine in the neuraminidase. If the neuraminidase mutated, that's in the active site, and the enzyme would be dead, so those viruses would have no selective advantage, obviously.
Right. So instead of just having something that sticks in there and that could potentially be blocked or ejected, if you have something that covalently bonds, it's really, really stuck, and the virus is not going to be able to get rid of it.
Is that like James Bond?
Yes.
Right. The way the BBC News writer put it, or actually quotes the study leader, Steve Withers, saying, "Our agent latches onto this enzyme like a broken key stuck in a lock, rendering it useless."
You know, I think you could tell people the way it is. If they don't know what a covalent bond is, you just... All right.
Well, there's limited space in media stories, and that actually is a pretty good soundbite explanation of what's going on.
I don't like it. I'm sorry. I mean,
You're just...
am I cranky today? I'm sorry.
You're just picking on this. Wow.
It's a good paper. I think it's very
nice. It is actually a really good paper.
But I just think we need to... anyway, you know what I think. These are 2,3-difluorosialic acids. So they make a bunch of derivatives which have all the required pharmacokinetic properties and affinity and so forth. There's a lot of chemistry. A lot of chemistry here.
Yeah.
Half-lives are important for these things. And then they start to look at them. They first start with neuraminidase enzymes, just the proteins, and seeing how these things are inhibiting them. And then they finally look at them in cell culture and then in mice. So the compounds are good at inhibiting virus multiplication in cells. Let's see where the figures are all the way at the end here. No, I missed it. I can't find the table where the figure is. It past the mouse figure? Are they all out of order? Yeah.
The tables are afterward. Yeah.
Yeah. The table shows the various derivatives that they made, and they have 2 fluorines, and they're different conformations, and they have different side groups, and they're looking at the half-life of the inhibitor and how it affects a variety of H1N1s and H3N2 strains, and a couple of mutants that are resistant to the other neuraminidase inhibitors. So they find compounds that work comparably to the existing inhibitors. And they say there's no cytotoxicity. 5 millimolar is the highest that they go, comparable to the existing. And then they use a mouse model. So they have, by the way, they work on A and B strains, Okay, so A and B both have neuraminidase. They use a mouse model where they challenge mice with a mouse-adapted influenza virus, and they work pretty well there. They measure virus titer in the lungs. They measure the body weight, which tends to go down in mice as they get sick from influenza. And you can see that the fluorine derivatives are just about as good as zanamivir in terms of body weight and virus
Zanamivir
titer.
is
Relenza, which are disialic acid derivatives. And then we have percent survival. So actually, the fluorinated derivatives of sialic acids are as good or better than the zanamivir. As the lowest dose is 1 milligram per kilogram per day of the fluorinated one, and that is as good as 1 mg per kg as of zanamivir.
Mm-hmm.
And there's a huge amount of supplementary material for this paper, by the way.
It's a lot.
It's a lot of chemical synthesis.
Yeah.
Now, my
stuff.
friend, my friend Lynn Enquist would say, if they published it, that means they're not going to do anything with it. But this is not a company. This is, Simon Fraser University and some other places up in Canada and also down in Australia. CSIRO. So I don't know, but it's an interesting concept, right? So the idea is that this sialic acids, they have fluorines that bind the tyrosine in the active site of the neuraminidase. They make a covalent bond that lasts a long time. It doesn't break, so it inhibits the enzyme. And the mice don't report any side effects, but of course you really need to look more carefully and also in other animals to pick up side effects. So it's not clear. So I asked Peter Palese, who is the neuraminidase guy. I have to tell you this. Peter Palese was, I think he got his PhD in Austria. And what he did is he purified neuraminidase from pig kidney and characterized it. And then when he came to the U.S., he began to work on flu eventually, and then was interested in, he actually published papers on inhibitors of neuraminidase, and then he had this TS neuraminidase paper. So he very early on in the 70s was thinking about an inhibitor. And it was only till we got the structure that it could be realized. But he told me a story once. He used to go to the abattoir and get pig kidneys, and he'd put them in buckets, and he'd cover the buckets with cloths, and then get on the bus to go back to the lab, holding the buckets of pig kidneys on ice, I guess.
Wow.
Yeah. It's the old days. Okay, so I asked him what he thought, and he wrote, these difluorosialic acids are legitimate inhibitors. They are very similar to the old dideoxysialic acid inhibitors, which are ion covalent transition state inhibitors, and they bind directly covalently to the tyrosine of the active site of the neuraminidase. Thus, it is less likely that resistance occurs by changes in the neuraminidase. If the tyrosine target would be changed, mutated, then the neuraminidase would be inactive. The problem is that these highly active compounds will also bind to tyrosines or serines, etc., of other proteins, and thus are likely to be very toxic.
Well, these don't appear to be very toxic, at least in the mice that they looked at.
Well, it didn't kill them.
It didn't kill them.
But
right,
who
we don't
knows?
know.
I mean, maybe they have a lot of pain. Maybe they have internal injuries.
Right.
Maybe their liver enzymes are going through the roof.
Dizziness, dry mouth, yeah.
Exactly. Maybe they, if they went to an opera, they wouldn't understand it.
They wouldn't understand it, yeah.
So
maybe
these
they
things.
shouldn't drive a car or operate machinery.
So the point is that just because the mice lived doesn't mean that there are side effects.
Right.
And all of you who work in industry know this. So anyway, it's an interesting lead. But Kathy, apparently the press loved it, right?
Well, there were a couple of things that we saw. BBC News and then this other eScience News, which I think is just a web page based thing. But yeah.
Yeah, anything to do with flu gets news. It's a big deal.
Well, and the idea that these drugs might be less prone to developing, or the viruses might not develop resistance to these drugs. I think that's what's attractive.
I don't know if I said, but they did test viruses that were resistant to oseltamivir, and this drug worked against those. So that's good.
Right.
But obviously they'll have to do a lot of tox before they do anything else, and that's where a lot of drugs die, I'm told.
Yes, indeed.
In tox. or detox, as it were.
Yeah.
But anyway, that was nice. Tell your class that they were the inspiration for a TWiV.
Right.
And they'll say, what? What's TWiV?
No, a couple of them know anyway. I mean, they know from
Just
the
kidding.
beginning they can get extra credit by writing up old TWiVs.
Oh, that's
And
cool, yeah.
Whenever there's something that's a current TWiV, I'll tell them. So, yep, they'll know.
Current TWiV. That's a very catchy thing, current TWiV. Mm-hmm. Our next paper also comes via Kathy, and this is a paper in the American Journal of Tropical Medicine and Hygiene, one of the really cool titles of a journal. It's been around a long time.
Yeah.
Detection of Eastern equine encephalomyelitis virus RNA in North American snakes. And I'm so glad they put virus RNA and not virus in the title. Because they do in a few places in this manuscript say virus in the blood, and I will pick on that, of course.
Of course. Right. And I don't regularly read that journal. The way I found it is the article was covered in Microbe, which is the news magazine of the American Society for Microbiology.
I liked it.
So-
I think it was a cool paper.
Yeah.
Eastern equine encephalitis virus, EEEV, E cubed V. That's another way to look at it.
Oh, yeah.
Toga virus, enveloped positive strand RNA virus. It's a mosquito-borne pathogen. And apparently it goes from New England to Florida and as far west as Ann Arbor, Michigan.
They don't say Ann Arbor, but they do say Michigan.
Well, I'm trying to
I
make
know.
you feel good.
Right.
You feel fine. You don't need me to.
Right.
Michigan, they just say Michigan. Circulates year-round in Florida, but seasonal outside of Florida. So the issue here is that it gets reintroduced from Florida to the northeast U.S., and then it stays for a couple of years and then goes away. So how does it stay over the winter, right?
Right.
Because in the winter, the mosquitoes aren't flying around, right?
Right.
So, Alan, mosquito biology, since Dixon isn't here,
Uh-oh.
what do the mosquitoes do in the winter? Do they hide under the leaves?
I think the generation of eggs makes it through the winter or something like that, and it's probably in dirt and leaf litter.
Okay. So the one idea is that, there's virus in the eggs and it's transmitted to the next generation, but I'm not sure that has ever been established.
And I'm sure it varies from one species of mosquito to the next, but there may be some that are killed off by a hard frost.
So there's a great page at the CDC on Eastern equine encephalitis, transmitted to humans by the bite of a mosquito. And if you'd like to know how many cases a year there are, there's a cool page where they report neuroinvasive disease. So most of the time you get a fever, or achy kind of disease, and it goes away. But sometimes it gets into your CNS. And so from 1964 to 2010, anywhere from 5 to 20 CNS cases in the U.S. every year. So it's not a lot. It's pretty rare. And you can see the map where they've got these cases. And Massachusetts is included, Alan, but not Rhode Island. Not Connecticut. Sorry, Rhode Island is. I wonder if it competes with the Lyme disease, Just
or maybe
kidding.
it's just reporting differences.
And so it's pretty rare. And the thing is, this virus exists in a cycle that is between mosquitoes and birds. And the mosquitoes are Culicida melanura and avian hosts in freshwater hardwood swamps. Okay, and this Culicida melanura, sounds Italian, is not a vector it doesn't bite people. It feeds exclusively on birds. So they need another mosquito to bridge. They call it a bridge vector between the birds and the people. And these are Aedes and Culex species, etc. So the idea would be that those would bite a bird, pick up the virus, infect a human, and that would be the end of the infection because we don't make enough virus in our blood to transmit it. And sometimes they infect horses, which is why it's called Eastern equine encephalitis virus.
Right.
And Eastern, of course, I guess Michigan is now in the east, right, Kathy?
Yeah, in this division, I haven't looked at this map now in detail. I think it does not include the county where Ann Arbor is.
Is that right?
Yeah.
You're trying to
Just
get
misses
out
our
of this
county.
virus.
Yeah, right.
Do you have swampy areas in Ann Arbor area? Probably not. Right?
There are some swampy areas.
You have hardwoods?
Oh, yes.
Oh, yeah?
Yeah.
Okay. Well, you would fit.
Yep.
Anyway, back to this paper.
Which is by
Yes.
Bingham, Graham, Burkett, Cadena, White, Hassan, and Unash, some investigators from Auburn University and University of South Florida.
That's right.
We
University
didn't
of
read.
South.
Did we read all the authors on the previous one?
No.
No, but there are a lot of them, but I did mention Stephen Withers.
Yes,
Kim was the first author.
Kim is the first author.
And they're from Simon Fraser and UBC and other
Right.
places, right?
Kim Resende, Wennekes, Chen, Bantz, Buccini, Watts, Pilling, Streltsov, Petrich, Liggins, Barrett, McKim, Breshkin, Nikura, and Withers.
Right. Okay.
So what they tell us in the introduction is that they have, or others have found EEEV in certain kinds of mosquitoes that prey on ectothermic hosts.
Those are ghosts, right?
Exactly.
No. Cold-blooded animals.
Cold-blooded animals. And and then in particular, Culex teratans feeds on amphibians, and Culex peccator and Urinotinea saphinerea preys on reptiles. Wow, these are big names. I prefer poliovirus. So they said maybe the snake is the overwintering animal where this not an animal, reptile where this virus lives in.
Reptiles are animals.
Are they animals too? I'm just afraid that someone's going to yell at me for calling them animals. You just got to be careful here on TWiV. So these people went into the Tuskegee National Forest, which is in Alabama, I understand, and collected samples from snakes. Oh, they took an ml from each snake, from the caudal sinus. I guess that's something at the back end, right?
I guess so.
Caudal is back. Mm-hmm. 1 ml. And these snakes were, some of them were dangerous, right? Cottonmouths.
Yeah.
Those are poisonous, right?
Yeah, cottonmouth and copperheads.
Do you have, cottonmouths were the most common, and then had copperheads too. Do you have those out in Michigan?
I don't know for sure. I remember when I moved to Georgia learning that all the nasty snakes in America are in Georgia. But okay. I haven't heard that about Michigan. So
I know we had them. I think we had both of those in Maryland. I don't know if we have either in Massachusetts.
Oh.
So they took the serum back to the lab, and they looked for antibodies to the virus, and they looked for viral RNA by reverse transcriptase PCR. And they found, let's see, quite a few seropositive snakes, 54 seropositive snakes. How many did they collect in total? Anybody see the number here?
I'm looking at the table 1. It looks 60-ish.
So good.
Mostly cottonmouths.
Some others.
Microbe article says 150 snakes, so that might include controls.
And if anyone wants to know how they knew that they had already sampled a snake, they mark them. Mm-hmm. I don't know how you mark a snake. I guess you get a Sharpie and you write something on it. Put a band on it. A band would slip right off.
Probably some kind of a dye marker or a tag or something.
Maybe.
Somebody will tell us.
Yeah.
And depending on your survey period, it wouldn't matter, if it washed off in a week, if you're surveying them all in one go.
So 66 seronegative cottonmouths. One was PCR positive, and then of the 54 seropositive snakes, 12 were positive for RNA, viral RNA,
right,
which suggests that there's virus in the blood, which is what they sampled, but they didn't look for infectious virus, I believe.
They did, but they were unsuccessful. They tried to culture
they did try and culture
EEEV from their samples in this study, but they were not successful. And they thought maybe it was due to sample degradation from freeze thawing. multiple times.
Right. Oh, and I just wanted to back up and correct something that I just said. They didn't just sample at one time point. This was over. They got us a set of seasons.
Yeah, they had month 4, 5, 6,
Yeah.
7, 8, and 9.
Yeah, so Sharpie probably wouldn't work. And don't they shed
Sharpie-
their skins?
Yeah.
And
Yeah.
I don't know how you mark a snake. It's a good question.
Well, if the authors are listening, and it was over a period of 2 years.
Yeah, so they see a little peak in the spring, and then it goes down, and then in the fall it goes up again. So they're making an argument that maybe this relates to maybe the mosquitoes. These animals, these snakes, keep the virus over the winter, and then they get bitten by mosquitoes and transmit it back to birds, and then back to humans. Mm-hmm. But apparently, this is the first report of the detection of virus in field-collected serum samples, and I'm saying it's not, because you didn't look for you didn't get virus, right?
viral
Okay.
RNA.
Viral RNA.
Viral RNA.
They made a slip in the discussion there,
Yeah.
and I highlighted it with my Sharpie.
With your snake tagging pen.
And then another one at the very last sentence: the demonstration that wild-caught snakes contain EEEV virus in circulating blood. Blah blah blah.
Yeah.
So it's not virus. So guys, what do you think? You can't assume that there's infectious virus there, but that's obviously an important component of this work, right? So you think they should go back and try it again?
Oh, absolutely. Now that they know that freeze thawing is probably not good for these envelope viruses, they should, sample the snakes and immediately try to culture them, whether they're seropositive or not.
Yeah.
But that's a lot of work. But
personally, I think they should take some of this blood and deep sequence it and see what other viruses are there. I think that would be cool to know the snake virome, right?
Yeah.
I mean, if you're going to catch all these poisonous snakes, how do you? I guess there are ways to do it with sticks and all that, right?
Yeah.
Yeah. I'm sure the herpetologists on this paper would have had techniques for that, and maybe you can trap them too. I don't know.
If you get bitten by a copperhead or cottonmouth, will you die, or you can be treated?
I think you can be. They're not super duper extra special lethal, as far as I know. There lots of people get bitten by them and survive. So, and I believe there are treatments of various sorts. Maybe antisera and such.
So, by the way, if anyone cares, they had racers, copperheads, cottonmouth, Decay's brown snake, Midland water snake, plain-bellied water snake, ring-neck snake, and timber rattlesnake. And they all had antibodies. And then they looked for RT-PCR, and then only the copperheads and cottonmouths were RT-PCR positive.
And interestingly, I don't know, did we comment on this already, that the snakes were not... the ones with and without the virus, there wasn't a skew for the gender of the snake or for the body weight.
But that's the thing I'm going to pick on, because snakes don't really have gender. They have sex.
They have sex, yes.
So that figure should have just been labeled snake sex.
Yes, gender is a human concept.
Right. It's a sociological construct.
Right.
But yeah. So, but I did want to say also, in reference to the follow-up that we had at the beginning, where someone was talking about presenting negative data, these investigators looked to see whether there was any correlation with which snakes had the viral RNA or had the seropositivity, and they also looked at sizes of the snakes, and they didn't see any correlation, and they present all that negative data here.
Yeah.
Well, but that's actually, I think that's also kind of an important point in terms of what might be going on. I mean, as Vincent says, it's not virus, it's viral RNA, but it would be very interesting if you'd found, for example, that the snakes positive for viral RNA were smaller.
Yeah.
Maybe there's failure to thrive, maybe there's some kind of disease, that this is suggestive evidence that maybe the snakes are just hosting this virus and not harmed by it.
Yeah.
Right.
That's another question entirely, right?
Yes.
No snakes were harmed in this study.
But quite a few were annoyed, I'm sure.
I'll bet none of these authors listen to TWiV. But if one of you are listening, let us know and I'll send you a mug.
All right.
But I bet
and he does not mean a picture of himself.
No,
but
not a picture of
a
myself.
bug.
All right, that's a cool paper. I like that.
It is. It's very interesting.
Our last paper has to do with this new coronavirus, EMC, Erasmus Medical Center coronavirus. I'm sort of looking at this all the time because I get about every few days an email from Matt Freeman, who's all excited about this. He said, "This is my SARS," because he's got a new lab, and if this is a new virus, he could make his career on it.
Yeah.
So let's bring you up to speed. Actually, there have been a couple of updates. The CDC just published an update yesterday, early release March 7th, summary of what's going on. There have been 14 confirmed cases reported to WHO with 8 deaths so far as of this report. 3 of them were identified in the UK as part of a cluster within one family, and there's evidence that there was transmission within that family. And this is a virus that is believed to have been moving around people from April 2012. So they had some, I think, specimens from someone ill at that point, and they were positive because the thing, I think, broke in the fall last year or early this year. I don't remember when we first started hearing about it.
I think it was fall of last year.
Fall of last year. And these are largely individuals who have been in Saudi Arabia, in that area. They've traveled elsewhere. Some of them went to UK and got sick there, but the origin appears to be in Saudi Arabia. Is that correct? Am I saying the right thing?
Right. 14 people in Saudi Arabia, Qatar, Jordan, and the UK.
Right.
And I think the initial cases were found around the time of the Hajj, which is the pilgrimage to Mecca, which is how Saudi Arabia to the UK travel and all that figured into the equation.
Right.
Okay.
And we've talked about this before, and we've talked about the fact that it is related to bat coronaviruses. However, it grows very well in human cells. And I don't know if we mentioned that before, but certainly the paper today is talking about that. No bat coronavirus will grow in human cells, right? A bona fide bat corona without any manipulation. So this is unusual. This paper is in MBio. It's also from, let's see. Ron Fouché's on the paper, and of course the virus was isolated in his lab. But the PI is Volker Thiel, who is in St. Gallen, Switzerland. And,
right. First author is Kindler, then Johnsdotter, Moot, Hamming, Hartmann, Rodriguez, Geffers, Fouché, Drosten, Müller, Diekman, and Thiel.
This is a paper in MBio.
Maybe we should set a cutoff for how many authors we read the list of.
I think Kathy likes to read them, right?
Yeah, it's kind of fun.
She likes to.
So anyway, MBio is open access, so everybody can check it out.
Efficient replication of the novel human beta coronavirus EMC on primary human epithelium highlights its zoonotic potential. So I picked this so that we could pick on it because this is basically.
You're just really in a mood today.
No, I'm okay. I'm just fine, really. It's been a nice week and everything.
This is Vincent in a good mood. People.
I think we should be critical, not just say everything is great. I mean, this is nice work here, but they just take human respiratory cells, as you will see, and from that they conclude it has zoonotic potential. But we already know it's infecting people, right?
Right.
Highlights its zoonotic potential. The thing is, if this were polio, I would never get it published in MBio. If this were adenovirus or whatever virus Alan works on, the fact is that this is a hot new virus, of course, so it's getting published. And I don't mean to take anything away from the work, but these things happen in science. Anyway, this is a study where they take primary cultures from human donors of respiratory epithelial cells. And these are cells that they get during biopsy or surgery. Unfortunately, if you have a lung transplant, they can go in and take some of the cells out. All right. I think even if you have some surgery for snoring or something, they do things like this. And they put them in culture, and they can add things so that they become polarized, right? And they have a top and the bottom, which are distinct.
Yeah, and this is actually, it's kind of a cool culture system where the cells differentiate and form something that looks for all the world like lung epithelium, and it's got layers, and it produces mucus, and it's got all these characteristics that it's not just a tissue culture system that you like HeLa cells. It's actually really mimicking a lung.
Right. And they do a nice set of nice virology experiments. They infect these cells with the EMC coronavirus, and they measure by plaque assay. They have PFU per mil. They measure RNA. They look at the infected cells by immunofluorescence, and they can see, for example, that the virus produced. They have a nice one-step growth curve. It's kind of nice. And they say the virus is produced mainly from the apical side of the cell, which is interesting, right? Because that we think that when viruses are mainly released from the apical side of, say, the respiratory tract, that they're going to be shed and transmitted, but they're not going to be systemic. But these patients have some systemic symptoms. They have, besides pneumonia, they have kidney failure, and they die. So maybe cytokines are causing that or something.
Yeah.
One thing I found really interesting is that they took total RNA from infected cells and they do this technique called RNA-seq, where they sequence the entire RNA at high coverage. So you can figure out all the viral RNAs, mRNAs in principle, just by looking at the sequences, because each RNA will show up as an individual set of sequences. And I was just thinking how hard it used to be to enumerate viral mRNAs, right? In the old days, you'd have to run gels and do hybridization, and oh man, it's so easy nowadays. Yeah. It's really changed. So they identify all the viral mRNAs. What I found cool was that these cultures, when you infect them, they don't really make interferon in response to virus infection. And in fact, they use the EMC, they use SARS coronavirus, they use a more benign corona, 229E, that causes more of a common cold syndrome in people. And these don't induce interferon. So they suggest that these guys have some kind of interferon antagonists that do that. Because, in fact, if you treat the cells with interferon to induce an antiviral state, then these viruses don't replicate well.
Right.
So the suggestion is that there's some kind of antagonist. So one of those viral proteins, somebody's working on it, I'm sure, is an interferon antagonist.
Mm-hmm.
Maybe.
And one or more.
Maybe, yeah. So they conclude, our data depict the zoonotic potential of human coronavirus EMCC, which I would say, is already known because it's infecting people, right?
Right.
And can't you guys think of some animal virus that does not infect people but grows really well in some human cell? I'll bet you can think of some.
Put us on the spot.
How about a mouse adeno? Does it grow in human cells?
Not particularly.
Okay.
We haven't really exploited that, but I mean, yeah.
Anyway, there are examples, but so I think growing in a human cell, even though this is as a primary human respiratory epithelial cell, I'm not sure how much you can conclude from that.
So they could have made the title of the paper be "Efficient Replication of the Novel Human Beta Coronavirus EMC on Primary Human Epithelium."
That would have been...
Period.
Yeah, they could have done that.
And titles shouldn't be sentences, and so they would have solved that problem too.
And they could have incorporated the interferon results. I think that's quite interesting.
Yeah.
Mm-hmm.
And another sentence worth highlighting in their discussion, considering that there is not yet any study reporting the successful isolation of a bat corona, EMC differs compared to known bat coronas because it displays broad replication capability in diverse mammalian cell lines. So either it's by chance that happened, or by growing in people it's acquired that ability, because this is obviously an isolate from a person, this virus.
Right.
Mm-hmm. And we should say that we don't know where this virus came from. They haven't figured that out yet.
Right.
But I figure at some point they will, right?
Maybe.
What do you guys think of this EMC? Do you have any feelings one way or another? No?
I don't know. Just reading the CDC site, where they talk about, should travelers be worried, and they give a few hints of what to do, and
that's right.
But 14 people, and 8 of the 14 people have died. Yeah. So 13 of the cases were severe.
Of the ones who've come to medical attention.
Yeah. Correct.
Yeah, I should point out that the CDC also today just issued a travel advisory about what to do. If you go to that area and you develop a respiratory infection, you should see your healthcare provider.
Yeah.
Yes, 14 people is not a lot, but you obviously want to be on the lookout for any new virus that has potential to spread in the population. My feeling is that SARS spread much quicker than this, right?
Yes.
Mm-hmm.
You were going to say, Alan?
Well, and it seems pretty clear that this thing is transmitting human to human, at least in a couple of cases, which is another important factor to consider, because if it were just people were sporadically coming out of Saudi Arabia or Qatar with it, and then you might say, well, each one is a unique spillover event. But this thing does probably have the potential to go person to person.
Yeah, I think so. It's all a matter of many things: how much virus is being made, how well adapted it is.
And how severe the illness is in the majority of people who get it, and we don't have a denominator for that.
Right.
So, there you go. All right. Let's do some email. Let's go back to Ibrahim's. Remember that, Alan?
Yes. You said you were going to paste it from above, and you didn't, so I'll scroll back.
Yeah, sorry.
Okay. Next, I have a flu question that I think I'd like the answer to with the previous question. It would help me take my dose of virology for this week. Influenza virus particles are covered by spikes of HA with NA with contradictory functions. HA binds sialic acid receptors to allow the virus envelope to fuse with the cell and to allow for entry of the virus. NA is a sialidase that catalyzes the release of sialic acid both from cells and virus particles during virus release to allow for the release of virus particles from the infected cell. Everybody got that?
Yep.
Okay. My question is, why doesn't NA function during the entry of the viruses and end up preventing entry? Because NA can catalyze this sialic acid, i.e. if HA binds to sialic acid receptors on the cell surface to allow for entry, wouldn't it be that NA then comes into action, preventing this binding from being established? If the answer is that there are more HA than NA on the virus particle surface, then why at the time of release is NA successfully releasing virus particles from the infected cell when HA at that stage is still binding to sialic acid when it's leaving the cell? If my question's not clear, just neglect it. Now, we wouldn't do that. I'm a PhD candidate waiting in the queue to get my visa into graduate school to be allowed access to the land of thoughts and experiments. That's a visa, can also be called a Cardiff, I think.
Yeah.
P.S. The weather in Egypt, I would not tell the numbers because it would make some people feel bad, but it is always sunny and always nice. Thanks for the great show. Keep up the good work.
It just so happens that Kathy asked me this question not too long ago.
Because one of my students asked the same question about
common.
the incoming virus.
If
If it has this ability to cleave the sialic acid, then how can the virus HA attach to the sialic acid in the first place?
Why doesn't it cut its own hands off?
Yep.
Yeah.
So I have included a photo of a HA assay as evidence. This is evidence A, and we'll include that in the show notes. It's all about kinetics, because HA binds to sialic acids faster than NA can cleave it off. And you can see this in this hemagglutination assay. There are samples that are clearly positive, which means the red blood cells are coating the well because they're all linked together by the virus. But if you look at the low dilutions, you can see that there is no hemagglutination because there's so much enzyme there. After a half hour or so, the NA, in fact, does cleave off the sialic acid from the red blood cells, so the red blood cells tumble down into the bottom of the well. So this is why you have to read a hemagglutinin assay within a half hour, otherwise the NA catches up. So it's pretty slow. And genetically, we can see this also if you make viruses that have different affinities of the HA for sialic acid. Typically, the NA compensates. So if you make a poor HA binder, the NA has to get poorer too. Otherwise, it will cleave off before the virus can get in. Is that clear?
That makes sense.
Yep.
So it does cut off its own hands, just not quickly enough to make a difference.
Yeah.
Right. Right, yeah. So the key rows in that figure are D and E.
Yeah. Yeah, where it's mostly positive, but to the left, you see the first well, 1 to 4, is not positive.
Right.
So I used to do these, right? And you'd read them in a half hour. And then if you left them there and went back to them, they were all buttons, because the NA is working. Kathy, can you take Cedric's, please?
Cedric writes, I saw this article in the Wall Street Journal that summarized a study in which researchers used publicly available information to find the identities of research participants who, quote, anonymously donated their genetic material. Do you think stories of this kind will become more common as research about and the practice of personalized medicine becomes more common? Will this make people more hesitant to embrace later medical breakthroughs or less willing to participate in research projects? And he gives the link to the Wall Street Journal article and to a science article, which is beyond a paywall. Thank you all for the time and effort you put into the This Week In series. Cheers, Cedric. So I didn't get a chance to look at these two.
You probably know about this, Alan, right?
This was actually one of the... there's a genre of these studies. The unmask the data types of security testing. And that was actually something I was thinking of, I think, last week or the week before. We had a discussion about pharma companies releasing data.
Right.
And this is exactly the sort of thing that keeps people up at night when they're talking about, well, gee, how much data do we release? How do we anonymize these? In this case, you've got genetic analyses, and it turns out you can deconvolute the stuff that was released supposedly anonymously and come up with individual identities. And that obviously raises a lot of ethical and legal issues for studies that are going to do this in the future.
Yeah.
And this is, as I say, there's a whole literature of this where you can use computational techniques to if you've got patient histories or whatever other kind of information you have, you can piece things together. And it is surprising how little information it requires about somebody to identify that person.
So do you think this will make people hesitant to participate in these studies?
Depends on what they're going to get. We're still at the stage where nobody knows quite how useful this stuff is going to be. At this point, would I participate in a study that was going to sequence my genome and anonymize the data? I don't know. I don't know if those data would be useful to me, or if they would be prejudicial to an insurance company, or how they might be maintained. And I know Rich is participating in something like that. It would be interesting to get his comments on this too.
I would do it. In fact, but I'm the opposite. I think I'm really into this stuff, and I think I don't worry about my privacy, although I know there are issues. But my son, I just sent his DNA off to 23andMe to have a bunch of polymorphisms run, and you have an option when you do this to check whether the data can be anonymized and publicized. I said, yeah. I said, do you want this anonymized and publicized? And he looked at me and he said, okay. I said, okay. But I mean, I know there are implications for other people, but in terms of insurance and all that, but I think, it's important to do this, and we'll find things out if we have more people doing it.
Yeah, and I probably would. It's something I would have to discuss with my family, but it's something I would probably end up doing.
Yeah. Well, I would discuss it and then do it, sure.
Yeah. How about you, Kathy?
Yes, Kathy.
I'd be a little more hesitant at this point, I think. I don't need to rush into it.
I need to rush into it. Absolutely. I want to do it and want to see what's there, and everybody can see it. I don't mind.
Well, you're approaching Medicare age.
Anyway.
Hey,
that has nothing to do with it. That has nothing to do with it. I just like research, and I think I want to make.
I'm with you on that. I would want to support a project like this, but I do have some concerns.
They could sequence my genome. They could have my microbiome. They can put it up. They can put my picture on it. I don't mind. I think it's important, and to serve as a model too, if that helps. All right. The next one is from Robin, who says live attenuated. How about active attenuated? I don't know, Robin. I don't like infectious I don't like live attenuated, right? But how about active? I don't know if active is good enough. I'd say infectious attenuated. That's what I do. Next one is from Rocky. Dear TWiV question markers. Oftentimes you briefly talk about if a virus is alive.
Have we mentioned that?
More than brief. Kathy, have you been involved in any of those discussions?
I have.
Okay.
I have.
I enjoy. Yes.
Somewhere I documented some of the shows that it was talked about, but now I couldn't find that. But I have the from minute to minute, and so go ahead.
I enjoy hearing each member's explanation for their stance. I'd like to hear this go in the other direction, though. Take a beehive, for instance. Are the workers having no reproductive future alive? I assume you would, as anyone would consider a human with no children alive, and depending on your experience with children, perhaps even more alive. But all of your talks that go this way tend to having the definition of life be more inclusive, virus, prion, etc. I'm interested in what your thoughts are towards making the definition of life less inclusive. For these cultural modalities where not all members have a chance of reproduction, could they be excluded? The idea will certainly be shot down, but I'm eager to hear why. For the bee example, you'd likely say other members of the species can reproduce, but for colonial bees, is the colony the minimal sustainable unit? Wouldn't that be an analogous to, say, a yeast cell being composed of living and non-living organelles? Anyway, I'm currently working through a giant backlog due to iPod death. I hope to someday catch up to your thoughts on this. Thanks for the time. Love the show, Rocky.
Can an iPod really die? Is it
alive? Yeah.
I don't know.
Dixon's not here, but a beehive with no future is certainly alive. But
and I would say that the individual bees are as alive as our individual cells.
Right.
Yeah, but he's picking on us, saying that viruses can't reproduce on their own.
Right. An individual bee can't reproduce on its own, but its genome is the same as the genome that's being reproduced by the queen.
The bee is metabolizing. It's taking in things and putting out things and making ATP and hydrolyzing it and all of that. And virus particles do not do that.
Right. And I think the distinction here is the definition between living and non-living versus the definition between the boundary that you draw for what is an individual organism.
Right.
So a colonial organism of volvox moving through pond water, the cells in it aren't really individually functioning. They're functioning as part of a colony. Somewhere along that continuum from that, which is clearly one organism, to a beehive, which is either a society of individual organisms or one organism, you could probably try and establish some sort of a boundary and say, an individual organism is such and such. But it's kind of a similar difficulty.
I think the bee in general is living, but if there happens to be a problem with a hive or a human who can't have offspring, it's not that they're not living. They have some genetic reason for that, and that doesn't make them non-living, right? Because the organism, as you say, is still living.
Right.
So, I want to reemphasize that I think a virus is an organism with two phases. Let's say the virion, which is non-living, and then the infected cell, which is living. And I'm coming to grips. I am coming to be very comfortable with that, because the virus particle does nothing, but in the cell it's living. So there you go. I have the best of both worlds.
Okay. It's like light being waves and particles.
Right.
That's right.
Both.
That's exactly right. All right, let's do some picks. Let's start with Kathy. You have two of them, right?
So I have two kind of related things. The first is the Finkbeiner test.
Yes.
So my younger brother sent me a link to this blog, which is on a site called XX Science. It's the new black. Anyway, the Finkbeiner test was just proposed on March 5th by a writer whose name is Finkbeiner, in order to put in stories about women scientists the things that are important about them being women scientists, and to omit the things that don't really have anything to do with their scholarship as scientists. And so it's a very interesting blog post. She cites an article that was published in Science in 2002 about Vera Rubin, an astronomer, where 12 of the story's 24 paragraphs mention Rubin's sex or gender roles. And really, it was supposed to be characterizing her science. And so these include that the story shouldn't mention the fact that she's a woman, her husband's job, her childcare arrangements, how she nurtures her underlings, how she was taken aback by the competitiveness in her field, how she's such a role model for other women, and how she's the first woman to ... So that was a very interesting article. And then turns out that Nature has a focus this week on women in science. And this, at least some of the commenters on this blog post and several people that I've corresponded with, make the point that if the article is about women in science, then it's reasonable to have these sorts of comments. But if the article is just really profiling the scientist, then you wouldn't do that for a man in science. You wouldn't talk about his childcare arrangements and his wife's job generally. So that's kind of the litmus test there. So I thought it was an interesting pair of things. And then, as you pointed out, Vincent, it's the International Women's Day. So probably Nature did time their issue to that.
Yep. Yeah.
And when I saw this post about the Finkbeiner test, which was going to be one of my picks of the week on an upcoming TWiV, I thought back. I don't really do a lot of profiles, so usually if I'm talking to a female scientist, it's just because I'm going to quote her about some aspect of her science, and so none of this comes into play at all. But I have actually done, I think, a total of 4 profile-type pieces. 2 happen to have been women and 2 happen to have been men. And I think I met most of the standards of this Finkbeiner test just because that's the way that I write the story.
Right.
And in fact, I now that I think about it, I definitely mentioned family issues in the 2 of men.
Huh. Because
they were relevant to the story, but I don't think it came up in either of the others.
Yeah,
it's interesting. It's good. I appreciate that.
I think it's an excellent idea.
It's really good. Alan, what do you have?
I have something that just came out yesterday, I guess. And this is it's the kind of paper that we might do on TWiV, but it's more of an epidemiology type of thing. I'm not sure it would really support a TWiV discussion, but one of the issues with the flu vaccine is that it is very hard to measure its effectiveness. And we've talked about some of the problems with that. So this is a study that just came out. Some folks at Vanderbilt and the CDC who did a study on patients admitted to the hospital with influenza-like or respiratory illness. And they looked at vaccination history, and they were very careful to validate vaccination history. So in order to have counted as having been vaccinated, they had to be able to verify it, either at the clinic where you got the shot or at your doctor. And it's not a huge study, but what they found was that the vaccine appears to have a pretty significant effect in reducing hospitalizations, particularly in people over 50, which is exactly what we would hope to see. But this has been a tough question to get at, and I think this is an important piece of work in the right direction. And that just came out in Clinical Infectious Diseases.
Yep.
Actually, it came out February 28th. I'm linking to a press release that's from yesterday.
Yeah, there's a link to the paper in the CDC page.
And it is freely accessible.
Is it freely? Good. That's
yes.
good. Be curious to see what Michael Osterholm thinks about this.
Yeah.
Because he's usually critical or yes, he's critical of the efficacy of flu vaccines. Maybe.
Yeah, and as I say, this is a small study, and it's one study, and it would have been nice to have these answers earlier, but
I know Nick from SIDRAP listens.
Yeah.
And so, Nick, what do you think about this? Let us know.
Definitely, let us know.
Okay, my pick is threading the needle. TWiV goes inside a BSL-4, and I have to pick this because I want everyone to watch it, not because it's a TWiV, but I think this is a really unusual thing. We went in a BSL-4. Alan, me, and Rich went in with people from Boston University, Ron Corley, Elke Muehlberger, Paul Dupre, and we made this 1-hour video which shows you just what it's like. I think no one's ever done this, and probably no one will ever do it again, because this could happen only because it was under construction.
Yeah,
it was a cool experience, wasn't it, Alan?
It was really, really interesting, and I want to just publicly call out again the incredible effort that the BU folks put into accommodating us.
Oh yeah.
And obviously they want to get kind of their side of the story because there's some controversies surrounding this thing, but at no point was there any, swaying of what we were looking at, and I don't think anything came up where they said, "Oh, we're not going to tell you the answer to that," or "We can't show you that." They let us go everywhere, and I think they were very, very forthright and transparent about everything, and incredibly helpful.
Yeah, so our friend Ricardo from Portugal wrote a comment on the page. He said, "I can talk about details I would never imagine. My students will appreciate it."
Yeah.
So we did this. We went up. We started 9 a.m. one day. We went all day till 9 p.m. Then the next day we went back in till the afternoon. And then we captured about 6 hours of footage. And then Ray Ortega and Chris Kondayan over at ASM edited it. And they had it at one point down to an hour and a half, almost 2 hours. And they said, we have to get it under an hour. So it was really hard to do those last bits. And I ended up going down there for a day to DC to record some more stuff and help them edit it. And now it's about 55 minutes, I think. So it's a good length. It's actually interesting so that you don't know an hour has gone by. And I just think everyone should look at it. I just sent the link to Tony Fauci, actually, and said, spread it around NIH. It's good stuff. You paid for it.
That's right.
So I want to thank all those guys at BU, the guys at ASM, and you and Rich, too, for spending time. And this is a really cool thing that'll be around. So everybody check it out, please, because I think you're going to learn a lot. Who's been in a BSL-4, right?
Right.
You ever been in one?
Certainly, I learned a heck of a lot.
You hadn't been in one before, right, Alan?
No.
And Kathy, have you been in one?
Nope.
Was there a lady at your place that used to go somewhere else to do BSL-4 work?
Sonia Gerard.
That's right.
Had done things during her postdoc with Stuart Nickel at CDC, and she did go back sometimes to do that.
Because when I visited before this last time, I met her.
Mm-hmm.
Is she still there?
No, she's not. She's in law school now.
Wow.
Yeah.
BSL-4 to law school.
Yeah.
Oh. I remember she was telling me how she would go somewhere else to do her BSL-4 experiments. Mm-hmm. I just thought she was very brave, but having heard Elke say that she worked while she was pregnant and was very confident, it's really a safe place. Mm-hmm. I mean, I think she told us she had 2 pregnancies and she just kept working through BSL-4. So,
but
Kathy,
she wouldn't work in the BSL-2 during the pregnancies.
Yeah.
Because there's no containment.
Right.
Yeah. So, Kathy, is that something we can mention about her? Or is that does that not pass the Finkbeiner test?
About Elke?
Yeah, that she worked while pregnant in a BSL-4.
Oh, I think that's fine because it's pointing to how safe she feels in it. I think that's
right.
It's relevant to the story.
Okay.
Right.
I'm very sensitive now, as you
right.
Just having seen that article. All right. We have a couple of listener picks. One is from Steve, who is one of our transcribers. Brief discussion with Professor Joan Stites about viral RNA on the Naked Scientists, Naked Genetics podcast for February 2013, titled The Wonderful World of RNA. And Steve and others are transcribing like crazy. We have a bunch of new ones up there, including the Tony Fauci episode, so go check them out.
Excellent.
They're even transcribing for my other podcasts now. It's really cool. So you've got Steve and a fellow in New York, Frank, and Rafael, a doc in the Philippines. They're all transcribing together and working on this together. So anybody wants to join in with them, send us an email.
Cool.
Hey.
Ed had some multiple picks, Archiving the History of Molecular Biology. He has the press release from the Wellcome Trust Codebreakers Archive. And this is unprecedented access to the great minds who led the molecular biology revolution. So it's basically materials from those days which have been archived, and there's this page with all sorts of links to this stuff, which is pretty cool. And then there's a video, a couple of videos from Cold Spring Harbor archives. So this is all related to, I guess, the 60th anniversary of DNA structure discovery, right?
No.
I guess the Cold Spring Harbor archives have all sorts of things in them. It's a pretty neat place. When I was out there last summer, I saw the Hershey Chase blender.
Cool.
It's in a glass case. Were there any other picks that I'm missing?
I think you got them all.
There were 4, right?
It's listener picks. So one from Ibrahim. Did he have a pick? No. That was from Ed.
That was from Ed. And you got Steve's.
Steve and Ed. Those are the 2, right?
Right.
I don't want to miss any. Yeah, there we go. All right, that'll do it for TWiV 223. And you can find us, as usual, at twiv.tv and at iTunes. And you can help us if you go over to iTunes, if you haven't done so already. And leave us a rating there and give us a couple of stars. And that helps us to stay prominent. And do send us your questions and comments to TWiV at twiv.tv. Kathy Spindler is at the University of Michigan. Thank you, Kathy.
Thank you. I was thinking earlier that not only was this fun and I learned a lot, but I was stretched from the organic chemistry to the organismal level of the snakes for today. So it's pretty wide range.
I tell you, after doing TWiV for a while, you really learn more. We are now starting to revise our virology textbook. So we had a meeting at Princeton this week. And I tell you, I knew stuff I never knew before. I was just amazed that I knew some of it, and I could actually contribute to the discussion. So TWiV is a good resource.
I find it incredibly educational.
It is. I mean, yeah, you just read these papers and you read them with the idea of being able to explain them, which is a different level from just going through them for your own purpose. So I really like that a lot. Alan Dove is at alandove.com and on Twitter. Thank you, Alan.
Always a pleasure.
Of course, our other co-hosts are off playing. Dixon is somewhere in Europe, and Rich is in Oregon skiing or snowboarding.
Not anymore.
He's back?
He broke his collarbone.
Oh, no.
Serious?
Yeah. Yeah, he did say that he got the hang of the snowboarding, so mission accomplished in that respect. But he said he's got a sling and it's a-okay. He'll be fine.
Yeah, he didn't tell me because I had told him to break a leg. Nice. Yeah, last Sunday he had texted me. He said, I'm in Oregon. I'm going to go snowboarding tomorrow. Wow. Yep. All right. Sorry, Rich.
Did he break it snowboarding? Do you know?
I suspect so because, in fact, I'm not on Facebook with him, but somebody else related that the first day it said Rich 1 Mountain 0, and then the next day it said Rich 1 Mountain 1.
Oh, I hope he didn't run into a tree. That's embarrassing.
No.
Evidently, the collarbone is the easiest bone to break, or most commonly broken bone, or something. So,
sorry, Rich.
Yeah.
Get well.
Get well soon.
Get well.
But he's enjoying the snow and the vacation, I'm sure.
Yeah. I'm Vincent Racaniello, and you can find me at virology.ws. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.